right. Well, thank you all for coming. So I'm going to talk about entropy scaling search of massive biological data. This is joint work with, there we go. This is joint work with Bonnie Berger, William Yu, and David Danko out of Bonnie's lab at MIT. And I'd also like to thank Andrew Gallant, Michael Boehm, Jian Peng, and our funding sources. So, as you all know, BLAST is still the most common way to do sequence search. It's actually the most widely used computational tool in all of biology. And one of the wonderful things is that BLAST scales pretty much linearly with the size of the database. But the problem is that genomic data are growing exponentially. Now, so this is the number of sequences in GenBank since 2002. This is real data, so it's a little noisy. It's tempting to think that cloud computing is going to solve this problem, and indeed it might save some money and provide flexibility. I'll note that Amazon is here with a booth promoting AWS, and Google and the Broad Institute have teamed up to put GATK on the cloud. But cloud computing doesn't address the fundamental issue. First of all, as transmission bottlenecks tighten, and as analytics move to more remote places, field sites, etc., even getting data into the cloud presents a challenge. And moreover, cloud computing is still constrained by Moore's law. So genomic data are growing exponentially. This is a log scale plot of computing power and sequences in GenBank. So sequence data is on a bigger exponent than Moore's law. So this implies that running time is going to grow exponentially and becomes infeasible. Computers aren't getting faster fast enough to keep up with the data. So we need better algorithms. So a common strategy in computational biology is to exploit the structure of data. And people have exploited the structure of biological data in a lot of different contexts. Here are a bunch of approaches. I'm not going to go into them, but you'll recognize a lot of them. I'm going to talk about another approach for exploiting data. So one kind of structure we can exploit is redundancy. This is a phylogenetic tree of some fruit fly genomes. And basically what you're looking at is the red bar is the size of the genome of Drosophila simulans, the yellow bar, Drosophila seychellia, and the black bar is the size of the unique data when you merge the two, okay? So as we merge another genome in, we get a little more data. So basically what this is showing is that the unique data from all of these species is not a whole lot larger than, well, for all these species, not a whole lot larger than two of them combined. So Lowe, Boehm, and Berger introduced the idea of compressive genomics in 2012. Now, normally we think of compression as a space-time trade-off. We compress data, we save space, but it takes time to do that, and then we have to take more time to decompress it if we want to do any analysis. Compressive genomics is the idea of compressing data in such a way that we can perform computations in that compressed space, saving both space and time. So what I'm going to talk about here is three contributions. A formalization of compressive genomics, so really why it works and why we expect it to continue working, and an application to metagenomics, and an application to chemogenomics, or small molecule search. So first, let's talk about why compressive genomics works. So let's define approximate search as the problem of, given a database, for any new observation of the same type of thing that's in the database, which database entries are the most similar. So we can sort of visualize it like this. This is a cartoon of approximate matching, which is essentially what BLAST does. So we've got some queries and we've got a database. And approximate matching is coming up with a set of best hits for each query. So from the point of view of one query, we can look at it like this. And we can imagine our database is embedded in some feature space. So our query actually lives someplace in that space, but we don't have a coordinate system, so we can't say it's right there. And what we're trying to do is find all the points in that space that are within some threshold of our query. Naively, the complexity bounds on this are if we have m queries and a database of size n, approximate matching is on the order of mn, because approximate search is O of n. And the problem, of course, is that n is growing exponentially. So what I'm going to show you is how this can be done in something on the order of the metric entropy, which I'll explain, plus the output size times a scaling factor. And what's important is that in practice, this is much, much smaller than n. So here's the cartoon again. We've got our queries and we've got our database. Now, we can think of compression as, in a sense, clustering. And this is a cluster-based search. So we have some cluster centers. Let's leave aside how we choose them. And they partition the space. So from the point of view of our query, we can search over the cluster centers with some permissive threshold and some permissive radius, and we can identify the clusters that those centers belong to. So the centers within this radius, we identify their clusters, and we search those clusters with the smaller radius that the user actually wants. The threshold you would have given to BLAST. So far what I've described could be just like a grid search or a cell search. And normally this only provides a constant factor speed up. But that's an assumption based on thinking the data looks like this. And often the data really looks something like this. So many databases, including biological ones, have a much more interesting structure. Consider the case of genomes that have evolved over time. So this is sort of tree-like. I mean, it's not a tree, but it's sort of tree-like with a lot of branches. And if you zoom way out, they're one-dimensional locally. But there's diffusion due to mutations. So if you zoom in, each of these branches is thick. And so we cover the data with spheres or hyperspheres of some radius. Maybe that radius is roughly about the width of those branches. And we can do this with a greedy clustering approach. And the number of those spheres that we need is the metric entropy of the data. Now, because all the points within a sphere are close to one another, they can be encoded in terms of one another. So they're highly redundant. And the number of those spheres is basically a good approximation of the entropy of the whole database. Also, the data exhibit what is called low fractal dimension. If we so given a point and some radius around that point, if we look at the number, or if we look at the number of data points within that sphere or hypersphere or whatever it is, and we increase the radius, if we have low fractal dimension, we don't expect the number of contained points to increase that much. So how would we implement a search on this? Well, we're going to use a search tree. And here the spine of this tree represents the cluster centers. And so this is the spine. And each subtree holds the data points in that cluster. So first we do what we call a coarse search on only the cluster centers. I should mention cluster centers are still real data points. They're not centroids, they're centers. And we find some cluster centers and we do a fine search on their subtrees. So the time this takes must be the coarse search plus the fine search. Now, if we say k is the number of representatives, this is O of k plus fine search. Ultimately, and the proof is in the paper, using the tools of fractal dimension and metric entropy, we get to this thing, which, to give it some intuition, is the number of clusters, the metric entropy, for the data. And given two radii for search, coarse and fine search radius, this is a scaling factor that depends on the fractal dimension. And for the trees we have to look inside, we've got the output size. This is the number of points we expect to output for a given search. All right, so we've got this interesting observation about some data spaces, and I want to change gears a little bit and talk about some applications. So metagenomics is the study of whole microbial communities, which might be in an environment like soil or might be in the human body. So BLASTX, which is the standard tool for metagenomic search, translates nucleotide queries into all 6 possible protein sequences and searches a protein database for matches. Now, BLASTX can be quite time intensive, but recent approaches like RapSearch2 and Diamond use reduced alphabets and clever indexing to speed this up a lot. So Diamond in particular has been the fastest metagenomic search tool thus far, or at least the fastest with good accuracy. So we applied this idea of entropy scaling or compressive acceleration to metagenomic search, and in fact to Diamond. And we've developed MICA, Metagenomic Inquiry Compressive Acceleration. And MICA can do its coarse search. MICA uses Diamond for its coarse search, and then it provides a choice of using either Diamond or BLASTX for fine search. And as far as running time, this is a log scale, so we could fit BLASTX on the slide. MICA is 3,700 times faster than BLASTX on a American gut microbiome data set that we considered, making it about 3.5 times faster than Diamond if you use Diamond for the fine search. If you use BLASTX for fine search, it's a little bit slower, but it gives you more flexibility in terms of all the options to pass to BLASTX. And nice thing is it can fit into any BLASTX pipeline as a result. So that's MICA. And I should mention, it loses no accuracy with respect to Diamond. It has the same accuracy as Diamond, making it in our tests about a little better than 90% recall versus BLASTX. The second application is rational drug design, or chemogenomics. So in rational drug design, molecules are designed to fit targets, but this is very expensive. So it makes sense to repurpose old drugs for new targets. And doing that typically relies on high throughput screening, which is traditionally done with large arrays of compounds and cool robots. But this is still expensive, so computational or in silico high throughput screening has been done more recently. And the basic idea is we're going to consider a drug as a chemical graph. And if it's a graph, well, if we can compute the maximal common subgraph of 2 chemical graphs, then we have a distance function on them. So we can do approximate search. But the problem is, and I should mention one of the excellent tools for this kind of search is the small molecule subgraph detector from Janet Thornton's group. And so the typical database we might search is the NCBI's PubChem. But the number of molecules in PubChem are also growing at a tremendous rate. I'm not going to claim that it's beating Moore's law or is even exponential, but it is growing tremendously. PubChem now has 68 million compounds. So we applied this idea to small molecule search, and we developed Amylite, which basically, I'm going to leave out the details, but it clusters molecules in such a way that we can search on representatives. And Amylite is approximately 150 times faster than SMSD with, again, better than 90% recall. This is, again, a log scale. These times are in hours. Yes, 40,000 hours, searching the October 2013 PubChem, which was 47 million compounds. So to sum up, we've seen this idea of entropy scaling search, which can take advantage of the structure of data if we have low fractal dimension and low metric entropy. And as long as we can cluster according to some sort of distance function, we expect that as we accumulate more data, we expect to mostly fill in existing clusters and very rarely creating entirely new clusters. So this gives us some hope that we can cope with this explosion of data going forward. I should note we also applied these ideas to FragBag from Budowski Tal et al. 2010 for protein structure search and got about a 10x speed up with virtually no loss in recall. So we've seen MICA, which is for metagenomic search, Amylite for small molecule search, and ESFragBag for protein structure search. The software will be available shortly, once I remove the password protection, at this website. Source code's on GitHub. Preprint is available on the archive, although I just found out last night that this has been accepted for publication in Cell Systems. So thank you very much.
Thanks, very good talk. Questions?
Unfortunately, the compression is time-consuming. The NCBI NR database on a 20-core machine takes about 40 hours. The nice thing is that's compressing the whole thing, but we can do incremental updates very quickly. Sorry? Yeah, we supply a current, we're going to update at least quarterly, maybe monthly, the current NR database. So you can just download it, you don't have to run the compression. Good question.
Other questions?
Have you tried other tracing some kind of question or?
So it's a good question. We cannot get false positives with this approach. So the only thing to consider essentially is recall. Yeah.
Are there any others? If not, thanks for raising.